6 months of focus and hard work can put you 5 years ahead in life. As it's always said that difficult road will lead to beautiful destination. And so as in the case of CSIR exam, if you need to accelerate, you need to build higher on the pyramid. So here I am to talk about important topics of Unit 12, that is applied biology, and tricks on how to prepare for it. So hey friends, this is Riya Kilmani from Biotecnika. With no more delay, let's proceed on to the video. So this Unit 12, it is about applied biology. So if you are a CSIR NET aspirant, you may be targeting or preparing 10 to 11 units for your CSIR NET examination. So this unit, it is asked along with Unit 13, that is techniques in biology. So if you are simultaneously preparing for this unit, it will add a bonus into your preparation. As this is a small unit, so you can prepare it parallelly with some other units. So if you are really going for this unit, it will help you to provide more marks in examination and also helps you to improve the chances of getting a good rank in CSIR NET examination. As I've already mentioned that this is a small unit, so let's see what should be your plan, how to execute with it, what are the important topics and tricks to prepare for this unit. So now we'll see what the list of important topics. So the first topic over here is approaches and techniques for development of transgenic plants. So you all know that what a transgenic plant that contains a foreign gene or the transgene. So here also you need to focus about the molecular approaches that is needed to identify the integrity of the transgene inside the organism, whether it is plant, animal, or as well as prokaryotes. So the second and the most important topic over here is agrobacterium mediated gene transfer. So I've already mentioned that there are 3 different techniques of gene transfer in organism. First is physical method, the other one is chemical, and the third one is biological method. So under physical method, you need to study about microinjection, particle bombardment, electroporation, that is physical method. Under chemical method, you need to study about the calcium chloride method as well as calcium phosphate. Under biological method, you need to focus on the agrobacterium mediated gene transfer. So this is the topics you need to focus over here under Unit 12. That is done or it is used to utilize to transfer genes inside the plant cells. So what all things you need to study under this is: what is the genetic transformation in plant? How it is done? What are the sequence of steps? What are the structure and function of TI plasmid? What are the different kinds of opines that are present in the structure? And what is the function of virulence gene? This is the most important part under this because you may expect the question in match the following form, like what are the different functions of virulence gene? What is A gene? What is virulence G, B, C, D, E, F? So like this, you have to just recall and learn the function of all the virulence genes in one line. So now let's see other topics. So the topic number 3 over here is animal tissue culture and cell culture strategies. So here you need to focus on the cell tissue culture media. You should be thorough with the basic terminologies, that is cell culture, primary culture, secondary culture, passaging of cell, what is anchorage dependent, non-anchorage dependent, what is microcarrier culture system, macrocarrier culture system, and also the propagation and culture maintenance of cell culture, how the passaging is done, what are cell lines. Now here you need to focus on the examples of mammalian cell line such as BHK21, HEK293 as well as Chinese hamster ovary. What is their source and origin and what is its significance, where they are utilized for their research purpose. So the 4th topic over here is PTC. Here you have to learn about the protoplast fusion. What are the chemical agents that are required for the fusion of protoplast? Now what is protoplast? A cell without cell wall, it is called as protoplast. So here you have to learn about the agents, enzyme that causes the fusion of the protoplast as well as the somatic embryogenesis. What are the different stages of somatic embryogenesis? Like there are 5 stages: zygotic, globular, herd, torpedo as well as maturation phase. Again, the most important topic is molecular markers and their significance. Here you need to learn about all the molecular markers that are used for diagnosis purpose. So it is RFLP, RAPD, AFLP, ISSR, SSR, etc. So now let's coming to topic number 6. It is vaccine and mechanism of immune response generations. So we all know that vaccination, there are different kinds of vaccine that are given to boost our immunity. So it is attenuated vaccine, it is recombinant vaccine, DNA vaccine. So here you need to only learn about the significance, how they are prepared, and also the examples. What are the examples of live attenuated vaccine, inactivated vaccine, toxoid vaccine? So like this, you have to cover this topic. So the topic number 7 is molecular assisted mechanism of breeding in plants, animals, and QTL mapping. So this is the most important topic that is asked in CSIR and focused every year for the exam, that is molecular assisted breeding or simply mass selection. And the 8th is therapeutic application of gene therapy as well as stem cell technology. So now let's see what are the strategies and how to prepare for this unit 12. So the list of important topics I've already shared, so you can just go through the slides again. And now let's see what are the tips and tricks to prepare for this units. So the first trick over here is comparative chart. So once you are preparing for comparative chart, it will help you better understand the interconnection between things. So always keep it short and precise. So what are different topics that you can prepare or you can utilize this comparative charts for revision is the topics like phases of growth curve. Now we all know that there are 4 phases of growth curve, that is lag, log, stationary as well as decline. So if you're preparing notes for this, it will take a long time or more time. So what you have to do, you have to just prepare a comparative chart for this. You have to make a 4 different column like lag, log, stationary and decline, and you have to write all the significance of this. What are these phases called? What are different metabolites that are produced during this phases? So the second one is types of culture method used in ATC and PTC. As I've already mentioned, under ATC you have to learn about microcarrier, macrocarrier, like all these culture system that they are there. And under PTC you need to focus on somatic embryogenesis, protoplasm fusion, anther culture, haploid culture, ovary. Like question from this, it is asked like what should be the culture method to generate the virus resistant plant? So it is haploid culture. So here you have to focus on the culture methods and its significance. And the third is ratio of phytohormones in root and shoot development. Now we all know there are 2 different kinds of cytokine that are involved in root as well as shoot development. It is auxin as well as cytokinin. So you have to learn about the ratio, whether it is high cytokinin to low auxin, it will result into shoot formation. So like this, you have to learn the ratio of phytohormone that are required for root and shoot development. And the 4th is types of stem cell and its example. Again, you can just prepare a comparative chart for different kinds of stem cell and the examples. So simply the direct question can be asked, what is the example of pluripotent stem cell? And over there you will find a 4 option where you have to select a suitable option for the type of stem cell that is being asked in the exam. So now let's see the tip number 2 is tabulations. So what all topics you can learn with the help of tables. So if you are preparing this table, it will help you to prepare a concise notes of all the topics. That will really help you to speed up your revision a day before examinations. So now let's see what are the important topics or topics that you can really do and prepare with the help of tables is enzymes used in somatic hybridization. What are the different types of enzymes that are used for somatic hybridization and embryogenesis? What are the different type of molecular markers that are used for diagnostic purpose and their significance like RAPD, RFLP, SSR, ISSR, AFLP? What are the different combinations of vaccine like DPT, MMR? So you have to just prepare a table for it. What are the different types of somatic embryogenesis? What are cell lines and its examples? What are the different classes of antibiotics? Like there are some antibiotics that are used to inhibit the cell wall formation in case of prokaryotes or bacteria. But some, they inhibit the DNA gyrase, some inhibit the protein synthesis as they will block the translation process, either elongation or termination step of protein synthesis. So what are the processes that are involved in phytoremediation? Like you have to focus on the simple definitions like what is phytoextraction, what is phytostabilization. So all these processes you need to learn in the form of table. What are the supplements that are used for ATC media and their respective role? So now ATC media means that is used for the culture of animal cell, because here we are focusing on ATC media. So it would contain carbon source, protein source, lipid source. So you need to focus on the function of all the fibronectin, whether it is glutamine, what is the role of glutamine, what is RPMI media, what all it contains. So here you need to focus on the media supplements that are used for culturing animal cell. Now the next is function of virulence gene. As I've already mentioned, you need to learn about the function of virulence genes. What is A gene? How the integration of transgene is happening in agrobacterium? It is with the virulence F. So you need to learn about all the functions of virulence gene. And the last is what are the agents that are used in protoplast fusion. So here you can see the tabulation for the antibiotic target. As I've already said that cell wall formation, there are different kinds of antibiotics for protein synthesis. Some other antibiotics can be there. DNA duplication will be inhibited by quinolones as well as folate synthesis. You can just prepare a table for it. If you don't want to prepare a table, you can just go for diagram. Here you can see a beautiful and visual representation of this tabulation. It is done with the help of diagram. You can just prepare a diagram for it and mention all the antibiotics and the functions of it or its target. Now let's see tip number 3, retain all principal formula. All the formula should be at your fingertips. You should always practice all these formula so that you should not forget a day before examinations. So what all formula you need to prepare under unit 12 is growth rate kinetics. Here you have to learn about the growth rate kinetics for animals as well as microbial cell culture. Now second is specific growth rate, that is mu is equals to 0.693 by generation or doubling time. And the third one is calculation of colony forming unit. So this CFU calculation, it is asked every time in GATE life sciences or biotechnology. So you should not skip all these formula. Now the fourth one is creating mind maps. So what is mind maps? Mind maps always help you to interpret things better. So it will give you a concise way of representation or of a detailed explanation of a topic at just one place. So always keep it handy and precise. So what all topics you can study with the help of mind maps is like methods of gene transfer. Now I've already mentioned there are 3 methods of gene transfer: physical, chemical and biological. So you can just beautifully explain all these or represent these processes in just a single place. And under biological we have agrobacterium mediated gene transfer. You can also do it for bioremediation. Just like phytoremediation, there is bioremediation that is used with the help of microorganisms. You can do it for all the processes that are involved under this process. And also the type of bioreactor that are used for the mass production of metabolite that are expressed or formed during the primary and secondary phase, like during the log phase as well as the stationary phase of the growth curve. And this metabolite, it is called as primary as well as secondary metabolite. Now here you can see the concept of a concept wheel that is designed by the Biotecnika itself. Now you can see how beautifully it is expressed. Now this is for agrobacterium mediated transformation. You can see the name of a topic is in the middle. You can just write it in the middle and you can see it is surrounded by all the important topics or theory that is related to it. So if you want to know more about the agrobacterium mediated process, how it is to be done, so this is just a theory. But if you wanted to know about the sequence, you can just visit the description box. The link you can find in the description given below. Just download the methods for it, learn it and recall, and that will be really helpful for your exam purpose. Now let's coming to graphical question. Now here you can also expect a graphical question under unit 12. Now what all topics are there where you can expect a graphical question? It is related to the functioning of the biosensor. Like they will give you a graph and they will ask you the linearity or sensitivity of a biosensor. So here you may expect a graph based question. So while attempting for the graph based question, you should be very careful about the x and y axis matrices that is being asked. So here you have to focus on the x and y axis, what is being asked in the examination. Also you can do it for growth curves such as geometric as well as exponential growth rate in plant, animal and microorganism. What are the type of graph that is formed as exponential as well as logistic growth curve that is in animals as well as plants. So now next is sequential representation of process. Now I've already mentioned that agrobacterium mediated gene transfer, it's a long and hectic process. But if you want to summarize it in a single page, how you will do it? You can just write a small and principal steps that are required for the process. So you can do it for the monoclonal antibody production, how the monoclonal antibody is to be produced, what is the sequence that is involved, what are the steps behind this, what is hat selection of this monoclonal antibody, and also you can do it first biosensor working mechanism, how the working mechanism of biosensor is being done. So now let's see how to approach this type of question. Now we have already seen what are the important topics, how to prepare for this topic with the help of charts, tables, whether it is mind map, graphical study, or in the form of graphs. Now let's see the very simple question that is asked in CSIR net December 2018. That is match the following questions. So how you have to approach to this kind of question. Now let's see. Here it is given column 1 and column 2. Here you have to match column 1 and column 2. But if you see the option Q, that is given antibodies. And you all know how to represent antibody. It is represented as capital A, small b. So where you can find this antibody in the column 2 options. So it is given in the first option, that is enzymes. That is monoclonal antibody as enzyme. So you can clearly match this antibody option Q with option first. That is where you can find Q with option first only in one option. So you can see only by picking first option or one option you can reach to the final answer, that is option B, P with 2, Q with 1 and R with 3. Now you may be wondering why I'm not studying or reading other options, because it is not necessary. If you are finding only one option as a right answer, you can directly choose that option as a final answer. So now I hope I have cleared all our doubts. What would be the important topics? What would be the plan and tricks on how to prepare and how to approach to questions. So if you like the video, do not forget to like, share and subscribe. Also share to your friends whosoever is preparing for this upcoming CSIR net exam, so that they can also get a list of important topics and tips and tricks out there. So thank you everyone for your time and patience. Meet you in the next video. Till then, bye-bye. Take care.